All right, well, hello ladies and gentlemen. What I'd like to do today is start by looking at this idea of evolutionary history with all life, focusing on the major taxa or the domains. Now, before we begin, I'd like to put emphasis on these big ideas. First of all, how do we organize these big groups, these major taxa, and then what are some of the fundamental features or key features of these groups, okay? And also as a caveat, I would like to make a point that, okay, the ideas that we're putting together here, or I guess the juxtaposition of these groups, is for the sake of explanation. You know, phylogenies aren't going to be exact in the way things are put together here. But again, just focus on the big ideas of the trends, okay? Now, the major distinction between the forms of life focuses on the nucleus. Prokaryotes, pro meaning before, kary meaning nut, or nuclei referring to nucleus, they lack a membrane-bound nucleus. These organisms tend to be more primitive. They lack membrane-bound organelles, obviously not having a nucleus, and they're the first forms of life. Archaea and bacteria are both prokaryotic, so the first forms of life, as we know it, were prokaryotic. Now, eukaryotes, on the other hand, have a true nucleus or true center. These are much more complex cells. There are membrane-bound organelles as being the beneficiaries of endosymbiotic events. Eukaryotic cells have all sorts of different organelles that can function in various capacities. So eukarya makes up the third domain, and many of the kingdoms that we're familiar with fall under the eukarya umbrella. Now, we think of life as being sort of this ring where there's this common ancestor to all life, and early on it gave rise to prokaryotes and eukaryotes. Now, a division occurred where a nucleus was created to create eukarya, but ultimately we can branch back and look at these relationships in some different ways. Now, the common ancestor for all life we can assume was unicellular. Evidence for that being the fact that archaea and bacteria, the first groups, or the first taxa, are unicellular as well. Likely were heterotrophic. There's no biological evidence or genetic evidence of autotrophic metabolism. And certainly we can assume that this universal common ancestor had DNA as its means of storing and transmitting genetic information, because all descendants of this particular organism uses DNA. Now, bacteria. First of all, how do you know a bacterial cell when you see one? Well, obviously they're prokaryotic. They lack a nucleus. They are unicellular, and another distinguishing characteristic is their cell walls. They have this peptidoglycan. So peptide refers to the peptide bonds between amino acids. So they're proteins amongst these sugars, gly meaning sugar. So you have these cell walls that are a juxtaposition of proteins, or at least polypeptides, and sugars. Now, when focusing on the anatomy of a bacterial cell, there are three basic shapes of bacteria. The spirochetes have this sort of obvious spiral shape. Bacillus has a rod shape, and this is a typo here. It's not bacillus. This is coccus, C-O-C-C-U-S. These are sphere-shaped bacteria. So, spirochetes, just think spirals. Coccus, the coccyx in your spine, at the base of your spine, is thought of to be like a berry. So with coccus, I just think of like a little berry. So they're circular or spherical. You can also distinguish them by their colonies. Worry about that when you take a bacteriology class. Don't worry about that for this class. Now, the basic anatomy of a bacterial cell is pretty simple. You have a single circular chromosome with no nucleus. You have these small circular plasmids. Certainly there's a cytosol. You have ribosomes to make the proteins. Everything needs ribosomes. You have a cell membrane. All cells have a cell membrane. You have the cell wall. Certain bacteria have capsules and flagellae, and they may have pili, or these little hairs. Now, these peptidoglycan walls can be arranged in a couple of different ways. The wall can be on the exterior of the cell membrane like we saw in the diagram in the previous section, or it can be sandwiched in between two membranes. Now, if the cell wall has this peptidoglycan layer outside the plasma membrane, then what will happen is it will take up a violet stain and stain purple. So these bacteria are referred to as being gram-positive. So the easiest way to remember this is just think positive is purple. So gram-positive cells, or gram-positive bacterial cells, stain purple. Gram-negative bacteria, because the peptidoglycan is covered by another membrane, do not take up the stain, so they appear, I think of them as being red. So let's think positive is purple, and then the negative would be red. Okay, so extreme close-up. Let's see. Again, positive purple, and gram-negative will be red. Certain bacterial cells can have capsules as well. This can be issues because certain capsules can produce toxins, not good for recipients of disease. All right. Now, bacterial cells lack membrane-bound organelles, but they still need to carry out metabolic functions. So it turns out that the membranes carry on majority of the metabolic functions for prokaryotic cells. So membranes do some of the work that other structures might in other organisms. For instance, cellular respiration will occur in membranes. There can even be photosynthesis that occurs in these extensively folded membranes of bacterial cells. So again, they have to make stuff, they just do it simply. Flagellae, or flagellum, is a whip-like tail. It's interesting to have these little motor proteins that use ATP, and it spins the tail so it becomes sort of like a whip. Helps bacteria move. And then pili are these little hair-like projections that can, again, be used for adherence. They can also be little tubes that genetic material can be released from. So the way I think of this, again, it's sort of like shooting spider webs out in different directions. So as a child, when I pretended to be Spider-Man, I didn't think I'd become an adult that imagines spider webs being shot out through bacterial cells. But anyway, think of little hair-like projections. All right, genetics, you're familiar with all this stuff. Okay, you know bacterial cell has a single circular chromosomes, and then there are extra pieces of circular plasmid DNA as well. Sure enough, they produce ribosomes. Bacterial cells need to make proteins, just like any other organism, so they do have ribosomes. Just so happens that they're smaller than eukaryotic cells. Now, in terms of reproduction, bacteria reproduce asexually through binary fission. So bi is 2, fissus means a split. So bacteria typically copy their genetic material and divide in half. How quickly? Well, under ideal conditions, every 20 minutes. So don't let your food sit out too long before you put it in the fridge. All right. So there we see, DNA is replicated. You have that origin of replication. Replication occurs in two different directions. You wind up with 2 circular chromosomes, and boom, 2 bacterial cells. Now, bacteria can also exhibit horizontal transfer. What they can do is pass DNA back between bacterial cells, and there are a few different ways bacteria can transfer DNA that we'll look at here shortly. The first is transformation. You did this in the lab with the jellyfish gene. You can insert plasmids into bacteria. When you heat shock those bacteria, loosen up the cell walls, and whoop, plasmids go in. They get new genes. Griffith did this as well by allowing bacteria to take up genetic information just to show it helped in the process of determining that DNA is what gets passed on and transforms cells. Now, bacteria, like what we saw, excuse me just a second here. All right, sorry about that. Had a student stop by. So again, you can have bacteria take up genes through transformation. Also conjugation. To conjoin means to bring together. So those little tubes, the pili, can actually join between bacterial cells, and then they will actually transfer DNA between the 2 cells. So here they show, yes, there's a plasmid, and it gets transferred through this pilus to another cell, or some of the actual main chromosome can be passed over. Portions of that can be passed over as well. There's also transduction. We saw that when we looked at bacteriophages and how they could insert different genes into bacteria. So that can transform the bacteria as well. So, you know, we're familiar with how all that happens. Let's see. Now, bacteria do evolve quickly, which is sort of counterintuitive since we know they divide asexually. But they do have relatively high rates of mutation because they lack the machinery that eukaryotes possess that fixes errors in the DNA. So errors can accumulate relatively quickly in bacteria to allow them to continue to survive. Additionally, any mutations that occur in plasmids can be passed back and forth between bacteria. Beneficial genes can be passed through a population relatively quickly. Additionally, with that quick generation time, bacteria can pass along mutations to descendants relatively quickly. All right, well, big idea, bacteria are everywhere, right? You see them just about in any environment. They basically are either autotrophic or heterotrophic. Let's see, some of the first life, particularly early bacteria, were cyanobacteria, these blue-green bacteria. Now these organisms are significant to the history of life on Earth because they began to change the composition of gases in Earth's primitive atmosphere. These photosynthetic organisms began to actually pull CO2 out of the air and pump oxygen into the air as a waste gas. And that opened the door to all sorts of ecological changes that led to evolutionary advancements leading up to us. So big stuff. Again, there's a species that can live in nuclear reactors, so they can handle all sorts of extreme environments. This just recognize for now that we need nitrogen in organisms to produce proteins, and bacteria can take nitrogen gas from the air and convert that nitrogen gas into a usable form of nitrogen that plants take up, and then we eat the plants or eat the animals that eat the plants. So bacteria are critical to the survival of eukaryotes because they take nitrogen out of the air to help with the production of proteins. Visually, you know, quite stunningly, you can see bacteria as well in the radiation that they can emit in bioluminescence. So the anglerfish in Finding Nemo actually glowed because of bacteria. Let's see, bacteria, despite being beneficial or non-pathogenic in many cases, can cause disease in specific organisms. So certain bacteria, you see the various species there, certain bacteria can cause specific disease. So, you know, E. coli and Streptococcus are a couple of familiar ones along with Listeria. So various diseases caused by different bacteria and where they can attack the body. Oops. Just to go there. Lyme disease is caused by the deer tick, which passes along a bacterium, and the bacteria can affect your central nervous system. Now, we can fight bacteria using antibiotics. Certain substances can begin to break down cell walls in bacteria, so it can attack those peptidoglycans. Additionally, you can go after the ribosomes that make the proteins. You can try and attack the DNA. So there are various means of sort of going after bacteria to slow their growth. And penicillin is a great example of that. Here you can see this mold, and the chemical produced by the mold creates this zone of inhibition because it affects the cell walls of the bacterial species. All right, so there we have it, bacteria.